So welcome to another Origin seminar today. Today we have a talk by Antonin Affholder, who did his PhD at ENS and the Observatory of Paris in France. He's now a postdoc at the Department of Ecology and Biology, and he's going to give us a talk about coupling interior and atmospheres to predict and assess habitability for exoplanets. So thank you. So I would like to ask everybody that's online to please turn off your monitors and your audio to save some bandwidth for people that have lower internet speeds. If you have any questions, please put them in the chat. Then I will or keep them to the end of the presentation. But if you have any urgent questions, please put them in the chat, and then I will ask them to Antonin. So please take it away.
Thank you, Sebastian. So as mentioned, I'm going to talk about what has been my main project during my PhD, and I'm continuing to do that here at the University of Arizona, although I also have other projects, so things are going a little bit more slowly these days on the astrobiology side of things. But yeah, I will try through examples from my work in the past few years to describe this rationale that we're trying to push with our team at the Ecology and Evolutionary Department. Use models from the theory of ecosystems backgrounds to couple with models of atmosphere and interior processes to assess in a quantitative manner habitability and biosignatures. Okay. Okay. So maybe a first point of entry into that world of biosignatures would be, what can we know about objects beyond Earth, exoplanets, but also planets or moons in the solar system? For example, for exoplanets, we might hope that one day we have those sort of spectra for the atmosphere of terrestrial exoplanets. It will help us understand what is the composition of their atmospheres in terms of molecules that are present, but also maybe their abundances. So yeah, for exoplanets, we might hope to know about the composition of the atmospheres. In the solar system, on the right-hand side of this slide here, I show Enceladus, which is a moon of Saturn that is an icy moon, but it has a subsurface ocean that sort of spews out in space in the form of those plumes that we see on the figure here. And those plumes were visited by the Cassini probe. I was able to measure a certain, to assess the composition of this plume for certain volatile molecules. And that can also be considered observable, that people might want to try to relate to the composition of the ocean of Enceladus and try to use that information to better understand whether the ocean of Enceladus is actually viable as a liquid water body to some Earth-like life, or if maybe it does contain a population of organism. So how do we go on and connect those observables to the actual candidate environments? For example, if we're talking about a terrestrial exoplanet, the environment would rather be a liquid water ocean. How do we connect those observables to those habitable environments, and what would we expect to see in those observables if those environments were actually viable to populations or were actually containing ecosystems? Again, so I will start with the example of Enceladus, which was the main object that I've studied during my PhD and for which we have publications that are out there. So not only does it have a subsurface ocean, but it is also likely to have hydrothermal circulation, so water being heated in the core, circulating in the core and being released at the bottom of the ocean in the form of hydrothermal vents. And then that warm water is assumed to rise across the ocean all the way up to the icy surface, icy shell, and be the source material of those plumes that we see in space. And in those plumes, the Cassini probe found interesting molecules, mainly dihydrogen, carbon dioxide, and methane. And dihydrogen and methane are typically evocative of not only hydrothermal circulation as a physical process, but also the chemistry that goes with it. The alteration of rocks, of minerals in the seafloor that is summarized in a set of reaction that we call subcontinization that produces H2 and methane to some extent. And on Earth, those hydrothermal vents, they exist and they are home to rather complex ecosystems that contain in a large fraction what we call hydrogen-trophic methanogens. So those, they combine the hydrogen and carbon dioxide into methane, and that reaction is their energy source that we call the catabolic reaction. So one might ask, well, if we find H2 in the plume, is that a sign that there is H2 being produced in the hydrothermal environments in sufficient quantities to sustain such a population of methanogens? Or maybe if there was a population of methanogens, wouldn't they consume the entire production of H2 by subcontinization so that we wouldn't see any of it left in the plume? And also looking at methane, could the methane in the plume be of abiotic origin, only from subcontinization that produces secondarily methane? Or could it be of biotic origin? Could it be produced by a population of methanogens? In order to answer that questions, we see that we need to really crunch the numbers and be quantitative to understand how much of H2 needs to be consumed by methanogens in order to survive, how much an established population of methanogens would consume of H2, how much methane would it produce, and for that we'll need a little bit of modeling. It's better at this, yes. But those ecosystems at hydrothermal vents are a little bit weird. They are very different from those ecosystems that surround us every day, on lands and in the surface of the oceans. Ecosystems are sustained by primary production ensured by oxygenic photosynthesis. So primary production is the way organic matter enters an ecosystem. It's the point of entry, the synthesis of organic matter from mineral sources. So on land, you have plants that get mineral carbon from the atmosphere, transform it into organic matter, and that's the primary production. But in those ecosystems in the hydrothermal vents, there's no light. So there is no light as a source of energy to ensure the primary production. And primary production is ensured by other reactions. One of them, I mentioned the methanogenesis, production of methane. And those reactions, so we call them chemoautotrophs, organisms that synthesize biomolecules from mineral carbon using those type of chemical reactions, mineral chemical reactions. And those typically yield low amounts of energy, orders of magnitude, for example, less than respiration, the oxidation of organic matter using the oxygen that we use as a source of energy is. So for example, for methanogenesis in standard condition, we are at about 30 kilojoules per mole, so barely enough to produce 1 mole of ATP, whereas respiration is in the order of million joules per mole. And so the importance of energy as a parameter of growth of this organism is often something that is overlooked in classical ecological modeling. To do that, we have to turn to literature from industrial processes, industrial methanation, production of methane, and those type of the non-standard literature to build a model of those organisms that will be suitable for us. And by the way, those chemoautotrophs that use low energy yielding reactions, they're not only in those hydrothermal vents, they're actually everywhere. They have a large impact on the current Earth geochemical processes, and they also dominated Earth's ecosystem for a long, long time before oxygenic photosynthesis was invented by evolution. So even in the context of looking at exoplanets, it might be interesting to be able to use a theory of growth and survivability for those organisms instead of using the classical ecological modeling for those regular ecosystems. But let's stay focused on Enceladus for now, if you may. And let's look at a pickle. It's not a pickle, it's a cell. Assume a cell surrounded by some resource being those regions from the catabolic reaction that produces energy. And the cell, with some machinery of proteins and stuff like that, is able to run the catabolic reaction and acquire the energy that is released from the catabolic reaction. We assume that the entirety of this energy acquired through the catabolic reaction is spent in synthesizing biomass. So we can write the balance of the energy in the cell, so the rates of the energy acquisition being yields ECAT times the rate of the catabolic reaction equals the rate of biomass synthesis times the energy required to synthesize 1 mole of biomass. And let's, for those organisms where we focus on energy, let's ignore problems that are associated with the access to those bricks that are turned into biomolecules. Let's assume that those organisms are in no short supply of those bricks. The only problem here in this assumption is access to energy. Then we account for loss of biomolecules due to, for example, denaturation that occurs at a certain rate that we call K maintenance here. So maintenance would be replacing those molecules that are lost, so that we can write the rate of growth as the difference between the amount of biomass that is synthesized minus the amount of biomass that is lost by denaturation, so that we can get the growth rate. Okay. And then going one step above, or taking one step back from being at the scale of the cell, let's look at a population of cells in its environment. So the catabolic reaction turns regions into products. We calculate the catabolic energy as a function of the state of the system. It depends on the temperature, on the concentrations of the different regions and products. And let's assume that those concentrations are changed, or their dynamics depends on the supply of a fresh medium to the environment that we're looking at, and the outflow, as if we were considering a well-mixed, continuously stirred bioreactor, and we put the population in that. We find that this template as a model to couple with larger biogeochemical processes is a useful one. We're considering a local environment, looking at what happens in it, but it is connected to the larger global environment and other processes through this inflow and outflow. And this model, the chemostat model, is well studied. Its dynamics are well understood. It has some nice properties, so it is very useful to us. From that, we can write the system of differential equations for the concentrations, CI, of regions and products. That is set by the chemostat dynamics here with the inflow and outflow, and produced or consumed by the population. Y here is the stoichiometric coefficients in the catabolic reaction, so it's either produced or consumed. And then the growth of the population, so the catabolic production of biomass minus the maintenance. And we also add a population level mortality rates. For example, cells might just die because they are untrained out of the system, or other sources that are not directly related to maintenance. And from analyzing the system of equations, we can draw a map of the conditions in terms of the energy as a function of concentration and temperature, I forgot to mention that those rates, the catabolic and maintenance rate, are expected to be functions of temperature as well, which is shown here. So as a function of the catabolic yields, the energy yielded by per mole of catabolic reaction and of the temperature, what are the conditions in the space that allow an initial growth rate to be positive? That is the orange area on this plot. So we have a sort of simple assessment of the viability of a system that is defined by its concentrations and temperature. And not only that, of course, but we can also run the dynamics of the system and predict the steady states of the system. And what we can predicts is that this is an inherent properties of those models, but the steady states of the system will be on this green line that is the limits of the viability area. So sort of by definition, an environment that is populated by a population will look like it is in scarcity of resource because the resource is consumed by the population. So that tells us that if you want to diagnose habitability of an environment, it is maybe not the best idea to just look at this environment that is potentially populated and look at the amount of resource in it, because a populated environment will look like if it is only barely habitable or not habitable at all, viable. But fortunately, most of the times in astrobiology, of course, we have access to observables that are not directly the environments in which we expect life to, or hypothesize life to be present, but rather things that are connected to the outflow or the inflow of these environments. And not only that, but also, that also tells us that a good strategy would be to look at the rates of production, the participation to biogeochemical processes that the population has, rather than try to diagnose the exact environment that is a candidate. Okay, enough with that. Let's go back to Enceladus and try to simplify as much as possible in a meaningful way the hydrothermal environments and connect it to the chemostat type of environment. What we assume is that hydrothermal fluid is released and at the seafloor and mixed with the ocean water. And it's released with varying intensity the further away we go from the center of the hydrothermal vent. Mixes with the ocean water. This gives a sort of a flat cylinder on the seafloor that contains various conditions with various mixing ratios between hydrothermal fluid and seawater. And those waters, they are warmed by the hydrothermal fluid, so they are buoyant compared to the rest of the ocean, and they escape this mixing layer and they compose this ocean plume that rises to the ice shelf. And there, in this environment, we can run our biological model. Given concentrations for the hydrothermal fluid and the seawater, we know the concentrations in the mixed layer, and we can assess whether a population of methanogens, of Earth-like methanogens, will be able to grow. And if it is present, how will it change the composition of this mixing layer and in turn the composition of the plume? To do that, of course, we need to set the concentrations in the hydrothermal fluid and in the seawater. We use constraints from the Cassini emission for some parameters, but mainly we use data from experiments of self-continuization. For example, here I show a distribution that we assume for the concentration of the hydrogen on the top, for the hydrogen in the hydrothermal fluid, and we just assume a uniform distribution between arbitrarily low concentrations, as if the potential for self-continuization in Enceladus was exhausted. Say if Enceladus is old, maybe the entire mineral stock has been already reacted with seawater. So assume low H2 concentration, or we are at saturation of H2 production in experiments of self-continuization, which is the upper bound here. For temperature, we use physical modeling performed by this study by Gaël Choblet of deformation of Enceladus interior and production of heat. Then, since we have distributions in, we have distributions out of simulated composition of the space plume. And that's what we see here on this figure. It's not exactly the concentrations in the plume, it's rather the rate of escape of H2 in the plume, technicality. And in magenta here, we see the actual observation made by the Cassini emission. So first good news is that our simulations, here it's only abiotic simulation. We just let the hydrothermal fluid mix with seawater and look at what happens. Well, those simulations are able to produce the H2 escape rate observed by the Cassini emission. Good news, but we can do more, of course, because we know in those simulations which one have conditions that are viable to methanogens and which one do not. So we can split this set of distribution, this distribution into two distributions. The blue one being those in which no viable conditions were found around the modeled hydrothermal vents for methanogens, and the orange sets is where conditions viable were found. What we see from this is the observations from the Cassini mission, they're really well explained by simulations in which viable conditions exist, not so much by non-viable simulations. More than that, let's take half of those simulations that are viable and let a population grow and see what happens. So that's the meaning of the green distribution now. And maybe surprisingly, it still does match the observation made by the Cassini mission. Maybe one might expect that it should be shifted further to the left to lower escape rates of H2, because H2 is consumed by methanogens. And it turns out that methanogens, that the concentration of H2 is higher where the hydrothermal fluid is in greater mixing ratio in the seawater, in the mixing layer. But that also means very high temperatures that are typically not viable. So there is a sort of trade-off where there's most hydrogen. It's also where there's the higher temperature. And so the population of methanogens is actually a little bit further away from the center of the hydrothermal vent than that. So that actually a lot of hydrogen can escape the hydrothermal system and find its way to the pool. So bottom line, what's the density units? Density units, it's a probability density. So here, if you integrate the blue plus the green plus the orange, you get 1. I hope that answers your question. So it has a unit that you can infer from the fact that it can be integrated to 1, I guess. But I'm not going to risk it. Okay, so H2 in the plume matches a populated hydrothermal environment. Then let's move on from the distribution for H2 and look at the distribution for methane. And here we see that the blue and orange distribution, so abiotic, meaning methane only produced by self-containization as a specific process, fail to explain the observations, whereas methanogens, they are actually able to produce observations. Conclusion, at least the basic conclusion is that self-containization is not sufficient as a process to explain methane. Well, let's entertain the idea, that there might be another source of methane, but this source is also putting methane in the hydrothermal fluid, not elsewhere in the ocean, just in the hydrothermal fluid. Maybe that could be pyrolysis of primordial organic matter in the core. We just don't know. But let's assume that arbitrarily there can be more methane. Now we are facing an ambiguous situation where both the abiotic and the biotic scenarios are able to explain the observations. When you look at the Bayes theorem in that context, what you can see is that whether the most likely scenario will be biotic or abiotic will depend on a completely unknown parameter, which is this fraction of simulations that are abiotic that we turn to biotic. I said earlier, I took half of those simulations and transformed them into biotic simulations. Well, why half? Why not 0? Why not 100%? This is actually an actual parameter that needs to be implemented in the inference process, which is in a way sad news because we have no idea what is the value of this parameter, but we can try to look at what would be its value so that the most likely scenario is abiotic, or what would be its value so that the most likely scenario is biotic. In this case, we show that it is around 30%, which is also bad luck because maybe people would have strong opinions if we situated these thresholds near 100% or near 0%. But since it's in that intermediate region, it's very hard to really say something further from that. But the framing of the conclusion would be a biotic scenario would be likely to explain the methane in the plume if the probability of life emergence, analogous to this parameter, is high enough. Okay, so we've looked at production of methane, consumption of dihydrogen, but what about production of other organics associated with life? What about, we're talking about the potential population of methanogens, but what would be its size? How does it compare to Earth's ecosystem? To that, we need to go back a little bit to ecological modeling, look again at the equations. And here I want to clarify something about ecosystems, is that there's a few quantities associated with an ecosystem, and they are not all the same, and they are not all related to biosignatures in the same way. For example, a basic metric for an ecosystem is the biomass stock. Basically, that is the amount of biomass in an ecosystem at a given time. Here, the given time being at the steady state when things are equilibrated. But the standing biomass would be you go on the mall of the campus, harvest all the grass, weigh it, and that's the standing stock, the stock biomass at a given time on this ecosystem. But that quantity is stationary at somewhere in the environment. It's local to the environment. So it's not directly related to biosignatures, right? This quantity is not exactly the quantity that we expect in the plume. It would be the quantity at the seafloor, at the hydrothermal vents. Especially so because methanogens in hydrothermal vents on Earth, they live in biofilms. They are fixed to the chimney. They're not free floating and going to the plume typically. There are cells that end up there. But in this type of modeling, in this theory, they would be counted as mortality. Cells escaping the environment is mortality for that model that describes the concentration of cells in a given place. But what would be something that ends up in the plume is rather what we call productivity. So it's a flux quantity. Here, well, we miss K-cats. That should be K-cat. So it's basically here the productivity in terms of biomass in general is the rate of synthesis of biomass at steady state. And it should be equal to the rate of mortality. And maybe those molecules that are denatured, they go out of the cell, they become diluted in the environment, and they might end up in the plume. Or cells that die, detach from the film, end up in the plume as well. Those are, more reasonable to connect to biosignatures. Okay, first let's take a look. Let's take a look at the potential size of our calculated population. So here we show a distribution where the solid black line is the prior case, so not making any assumption on whether the population is the source of the plume-found methane. But this is what we do for the dotted distribution here. Those simulations are the simulations that best match the amount of methane found in the plume. So just to say it again, this narrow distribution here, they're the subsets of simulations where the population produces methane in quantities that are the same that are found in the plume. And this population would typically have a size comparable to one single individual whale. So it's actually very, very tiny. That would be the entire mass of carbon in biomass for the entire Enceladus ocean. So very tiny. But in terms of productivity, here I show the production of biomass, of carbon biomass, per units of hydrothermal fluid. So sort of hydrothermal fluid specific biomass production. The vertical line, the vertical dashed line here is the best estimate for Earth's hydrothermal vents. What we see here is that the population that best match production of methane in the plume is also the population that matches well, to a factor of 2 actually, productivity of hydrothermal vents on land. So it's not that it's a weird population, very different from those that we have on Earth that is able to reproduce the observation in the plume, but it's really the typical hydrothermal vents, Earth-like hydrothermal vent that reproduces those observations. Okay, so that's for biomass and productivity, but in terms of biosignature, we've asked, let's assume that the mortality of the population corresponds to cells that escape the system and that might end up in the plume. And those cells, maybe a future mission will want to sample the plume and find a cell. What would be the required sample size so that at least one cell is confidently captured if there is a population of cells? Or in other words, what is the sample size required so that a negative detection is actually meaningful? That there is no false negative, that if the mission ends up not finding a cell, then it is a good evidence that there is no cell. All right, so that's what we plot here. On the x-axis, we have a concentration factor because we don't really know what happens while the plume, the oceanic plume, rises in the ocean. We don't know exactly what happens when this ocean material is transformed into the space plume. So accounting for this uncertainty, we plot the required sample size as a function of this unknown parameter. Again, the dotted line is assuming that the methane is biotic. So we see here that the sample size is actually reasonable in that case, if that's the hypothesis that we want to test. In blue, we have the orbit-under microscope design that is, in most instances, above this required sample size. But if we don't make assumptions on the origin of methane, then to reject the possibility of a population, we need much, much larger samples that can even exceed the entire sample size in the design of the orbit-under. And then the dashed line is really the 10% worst case, absolute worst case, and that also exceeds in some instances those thresholds. So it is possible to acquire this type of sample size, or at least I'm led to believe so by looking at mission designs, but it is definitely an ambitious requirement that we set here. And that's already assuming that cells are not completely destroyed when they're outcast into space, that we have techniques on board of those missions that would recognize a cell without ambiguity. So, yeah, that was for cells, but let's look at, for example, a molecule that is produced by organisms, glycine, an amino acid, but it's also produced by hydrothermal chemistry, it turns out. So here, again, as a function of this unknown probability that life emerges, right, that I was mentioning a few slides earlier, green is biotic production more likely, red is abiotic production more likely, and the y-axis is the glycine concentration. So what detection threshold should we aim for so that, again, we can reject with no false negative the hypothesis of biotic production of glycine? What we show here, this is in the posterior case, methane, biotic methane assumed case. In that case, 10 to the minus 5, 10 to the minus 7 moles per liter threshold detection is fine. I should have maybe put the worst case scenario where we show that 10 to the power minus 7 is really the safer threshold. And this is actually, if I remember correctly, something that people think they can attain with onboard instruments. So good news. Okay, just a very quick word. Recently, people have been discussing phosphate. Remember earlier when I was describing the model and I say, okay, assume that the bricks to build biomass are not a problem, they're accessible. For nitrogen, we had information that it was a good assumption because nitrogen is present in the form of ammonium in Enceladus' ocean. Also, methanogens typically are able to fixate mineral nitrogen, but phosphates, until recently, we didn't know much about it. Last year, there was this study that uses geochemical modeling and predicted that actually phosphate might be present in high concentrations in Enceladus' ocean. And even more recently, presented at the AGU in December, and probably is going to be published in a journal in the coming weeks, detection of phosphates in the plume, evocative of a very high concentration of phosphates in the ocean. But even if we didn't know much about phosphate, were we right about ignoring phosphate as a potentially limiting factor? And wouldn't a population of methanogens present for billions of years, potentially billions of years in Enceladus' ocean, would this population consume phosphates, the phosphate stock, until there is nothing remaining, no phosphate remaining, which would be incompatible with the with a recent measure. So let's just use the biomass predictions that we have, turn them into consumption rates of phosphates, and look at if it matches with those new results. So we found by analyzing that phosphate concentration in Enceladus ocean, even in the worst case scenario, really not likely to prevent initial growth. Also not likely to set the size of the population, the ultimate size of the population. And last, the population predicted being so tiny, the consumption rate of phosphates is also very small. And what we see on this figure here is that to change the concentration of phosphate in the ocean of Enceladus by even 1% would require, by our predicted population, hundreds of billions of years, maybe 200. So really not an issue, phosphate in that instance. Okay, I'll try to be quick now. I will just say a word about this other project that we have worked on terrestrial planets. I focus more on Enceladus for these zone examples because we have a well-established publication record in that matter, but this is more still work in progress type of work. But if we want to tackle exoplanets in the same way we have tackled Enceladus, we need, instead of assuming atmospheric composition to calculate temperature and look at the habitable zone that way, we need to let the composition of the atmosphere be a variable that evolves under the constraint of surface processes, of gassing, weathering, exchange with the ocean, etc. So we need to couple all of that together to predict atmospheric composition under different hypotheses and translate that into potential observables and try to build an understanding of exoplanet biosignatures and signatures of habitability from that direction. Just to give a very quick inventory of those geochemical processes that interact together. As I said, photochemistry, climate under the influence of the illumination from the star. Weathering of continents removes CO2 from the atmosphere at a temperature-dependent rate. So it is interesting as a thermostat. Removes CO2 from the atmosphere, puts it in the ocean. Seafloor weathering promotes, and so does continental weathering actually, precipitation of carbonates. So this carbon is transferred to the crustal reservoir, removed from the ocean atmosphere reservoirs. And carbon is also released by volcanic outgassing into the atmosphere. And the balance between those processes is one of the main drivers of the climates on a terrestrial planet. On a terrestrial planet, as far as we understand them. So we have painstakingly, is that a word? Couple all of that together. Especially challenging was the model of atmosphere. I think we really need a new generation of those models to couple with those approaches. But yeah, can ask questions about that. And then we ask what type of hypothesis can we look at in tests, right? So here I was talking about continental weathering and seafloor weathering, but all of that is really Earth-centric, right? It's what we have on Earth right now. And is the habitability of the Earth, has the habitability of the Earth through time been linked to the existence of all of those processes working together, especially this thermostat on climates, or is it expected to be a more generic property, this thermostat-like behavior? And that question is warranted because some planets do not have continents or continental crusts. The Earth is in an interior convection regime that we call mobile lid, where plate tectonics exists, continental accretion occurs, so there is continental material that can be weathered and participate to this cycle. But time-lit planets, such as Mars and Venus, put it simply, do not have continental crusts typically. So they are likely, if they have an ocean, to only have this seafloor weathering that I was also mentioning. They also have a different outgassing rate, and we might ask, whether those planets are habitable in the same way that Earth has been in their ability to maintain temperate conditions. And also, another legitimate question would be, are most terrestrial exoplanets like the Earth? And do they have plate tectonics and continents, or are they more like Venus and Mars not having continents? So is that something that we can try to infer from atmospheric composition? So yes, so we've run simulations in both those assumptions. So on top here, assuming that continental weathering can exist, so as a function of the installation, you call it, the steady state partial pressure of CO2 in the atmosphere. We see downward trends. So the more light energy is received, the lower CO2 is, as expected from how this thermostat, the continental weathering, continental and seafloor weathering thermostat works. Here at the bottom, we have planets in the stagnated scenario. And in this case, we only have seafloor weathering as a potential process to remove CO2 from the atmosphere, which is much weaker than continental weathering. And thus, a negative downwards trend exists, but is not as strong. This has some consequences, is that the planets that have continental weathering are actually more likely to be cooler when they're further away from the star than their stagnated counterparts, whereas the stagnated planets are more efficient to have a temperate climate at a low installation, but are worse at removing CO2 when there's too much light. So in a way that may be translated into habitable zones of those two types of planet being slightly shifted. But largely, the stagnated planets are capable of maintaining temperate conditions at the surface. Now let's assume that future telescopes will be able to look at the PCO2 in terrestrial exoplanet atmospheres. What would be the sample size and the quality of the telescope, the observational effort required to infer whether planets are in the stagnated scenario or in the mobile lead scenario? If we have only one data point, we won't be able to say much. But if we have more data points, and if our detection threshold is at the right place, then we can see appear some qualitatively significant differences between the distribution of only seafloor weathering planets that are typically remain at high CO2 values, even at higher installation, which is not the case for the continental weathering and seafloor weathering planets. So let's compare those two hypotheses. Either all planets are in the stagnated regime or all planets have plate tectonics, just to make it simple. And we also assume that there is a threshold of detection of CO2. If CO2 is above this, our imaginary telescope will be able to pick up the signal. If it is below, the telescope will not be able to pick up the signal. And plotting the required sample size as a function of the risk level that we're willing to accept, risk being risking to infer some evidence for the incorrect hypothesis. So we do that for either hypothesis and simulate sampling a lot of times to do that. So if the telescope is able to only pick up 10% or more CO2 in the atmosphere, then at 5% risk level, we need 20 or more candidates to be 20 or more planets to be analyzed. But at 1% and 1 per 1,000, actually it's pretty much the same. At 5% risk, we can be below 15 candidates and still infer correctly the good hypothesis. So we don't need necessarily crazy numbers of exoplanets to perform this type of inference, but we do need some, maybe more than we can get in the coming years, in the near future. Forgot to show this. And my time is completely up, which is good, because I will leave you with some bits of take-home messages of what we would like to do in the near future. As I mentioned, I really think that a crucial step is getting suited atmospheric models for this type of coupled approach. So not necessarily the best atmospheric model, but one that will be sufficiently good and run sufficiently fast so that it can be coupled in line with the rest of the model. also, those results about this hypothesis testing type of thing could be coupled with survey simulators, such as those used by Martin and Daniel. And of course, right now, I've only talked about completely abiotic things on exoplanets, but we would like to put ecosystems in those oceans and look at actual predicted biosignatures for those. And yeah, thank you.
Okay, thank you for your talk. Are there any questions? Oh, I already see that there are questions popping up in the talk, in the chat. So does the biomass stock or B star in your model is the same as the carrying capacity, K ecology?
So yes, in a way, carrying capacity, in general, when we talk about carrying capacity, it's to refer about models where the carrying capacity is an actual parameter. It turns out that in most models where we want to not look only at the dynamics of a population, but look at how it handles resource, there is no such thing as a defined carrying capacity as a parameter. It emerges from the model. But yes, it ends up being the carrying capacity. It defines the carrying capacity is a function of the environment in parts in those models. Yeah, it is defined. It defines the carrying capacity sort of a posteriori in those models where carrying capacity is not defined a priori. I don't know if that's clear or more confusing. So yeah, no, the very formal answer would be B star is the stationary biomass, and one might legitimately want to equate that with the carrying capacity, though the carrying capacity usually refers to a property of the environment rather than the state of the population.
Okay. Martin.
Yeah, I had a question regarding the plot you show, like stagnant lid versus mobile lid, and then you have Earth and Mars in there. Mars is stagnant lid, and Earth, as we know, is continent. So it kind of looks as if Earth, according to this simulation, would still be consistent with stagnant lid as well, to be expected, or is it just a tendency that's inherent to the model?
Yeah, difficult to answer that question with the data that we have right now. Though Earth has a carbon cycle that is a little bit more complex than what is in our model right now. The abundance of carbon in Earth's atmosphere is also largely determined by oxygenic photosynthesis, the emergence of plants that have trunks. Made a lot of carbon being stored neither in the atmosphere reservoir, nor the ocean reservoir, nor the crustal reservoir, but in those trunks. So that changes the situation a little bit. So the Earth, because it has life, does not compare too well with the simulations that we have. So that can be also a reason why.
I mean, of course, I like the idea of trying to do a population level inference if we can. Atmospheric measurements might help to distinguish between these planet types. You mentioned the idea that would actually mean that there are two outer edges of the habitable zone for these two different planet types. Can you think of a way of testing this idea? Like, is there a signature that we might be able to detect maybe only in the far future that would show us that there are actually these two planet types and they cause
that they both exist at the same time. So instead of considering that they are either all in one scenario or all in the other.
Right, we even have both planet types in one system in the solar system. So that's probably a simplification.
In only a single planetary system, and in this system, trying to assess which one is in what situation?
No, I mean, what you did was assuming either there are only these planets or only that planet. And we know this is not true because in the solar system we have already both. So that's clear, but I like that approach.
Yeah, no, of course, you can complexify the small perspective statistical analysis that I've done with varying the mixing ratio a priori between the different planets. And that would probably increase the number of candidates by I don't know how much. But it is definitely very easy to do from this, basically, maybe 2 lines of code. So yes, that's definitely something.
And maybe it's not possible for the individual planet, right? But on a population
yeah,
level, you can
exactly.
show that there is a fraction of stagnant. That's something that sounds realistic to me.
Yeah, well, it's realistic to calculate the number of candidates that would be required to perform that inference. Now, whether that inference is realistic or not, we need this calculation.
Yeah. Okay, let's chat offline.
Okay.
I saw that we have another question from Abel. Would you like to ask it yourself? You can unmute and.
Hi, everybody. My question is, can you hear me?
Yes, we can hear you.
Oh, yeah, perfect. Thank you. What are the sources of nitrogen in icy worlds in particular, and how that compares to other limiting nutrients? What would be the main source of nitrogen?
I actually don't really know the answer to the question. What would be the source of nitrogen in icy moons?
Yes.
I don't have the answer to that question. I only know that ammonium was found in Enceladus' plume, but then how would it be produced? I have no idea. Well,
Okay.
we have run some simulations of fermentation instead of methanogenesis in Enceladus' ocean, and fermentation actually produces ammonium.
Okay.
So as a biologist that only knows about biological things, I would say at least biology is able to produce ammonium. Yeah, otherwise I don't know.
Okay, thank you.
Are there any other questions? If not, then I would like to thank Antonin again for his talk. And then I hope to see everybody next week again during our next Origin seminar.
Thank you.